Hello, everybody. It's Phil Jacobus. It's October 6th. Thanks for joining me. I'm here in my office early today. It's in New York City. Today, I'm being joined by Peter Webner, who's going to tell us about a new biomarker. And he's a very interesting fellow. I'm sure you'll enjoy it if you can keep up with him. But first, let's take a look at several stories that are in our news today. We have a very interesting story about proton therapy. This is in the latest issue of our magazine because the new issue of our magazine is out and in the mail, and you can view it on our website. And .net has done a great job of covering proton therapy, and we have a great story this month, this issue that I'd love for you to read. Another very interesting story: Lafayette General and Auctioner Health in Louisiana are merging. These are two well-known organizations, and for them to merge is a big deal. It's part of this trend of consolidation. Now, before we get to Peter Webner, because he's going to talk about Cerianna, which is a new biomarker, I'm posting, as you see, a sort of disclaimer or a document that's required to be displayed. And I'd like you to look at this product safety and efficiency information of a newly FDA-approved drug, Cerianna. For full prescribing information, visit the website. Before Peter joins us, we're required to share that. It's great that I'm adhering to FDA regulations. And now let's go to our interview with Peter. Hello, everyone, and thanks for joining me, because today I'm joined by a very special person, Mr. Peter Webner, and I've known Peter for a long time, and he is extremely knowledgeable and creative and an outside-of-the-box thinker. Peter, thank you for joining us.
Thank you for inviting me. Appreciate it.
I've known you in many different positions, but currently you are the CEO of Zionexa. Am I pronouncing that correctly?
Zinexa.
Sorry?
Zionexa US.
Right, Zionexa US, and you're the CEO there.
Yes, I am.
And forgive me that I mispronounced the name, but
I'm harder to pronounce in French.
Yes, I'm a little bit. I have some knowledge about Zinexa, but do us all a favor and tell us just a little bit about Zinexa and what you're up to.
Absolutely. So Zinexa is a French-American company. We are a small biotech that are producing currently a tracer for breast cancer named Cerianna. We have several other products in our pipeline, some combination diagnostics and therapeutics, but our real focus today is our recently approved breast cancer PET imaging biomarker, Cerianna. It was approved in May of this year, and we expect to start launching it next year, January, using our commercial partner here in the US, PETNET Solutions.
So PETNET will produce the biomarker and distribute it and with around the markets, I take it.
Yes, we have an initial launch plan where we're going to launch out of 10 manufacturing centers. The first wave will be New York, Raleigh-Durham, Jacksonville. We are also going first wave out of San Francisco, Los Angeles. Shortly thereafter, Seattle, Philadelphia, Fort Lauderdale, Dallas, and Chicago.
I see. So you're covering a lot of markets. Tell me, how is Cerianna different than other products which are currently in the market?
So the unique thing for us is within breast cancer, the primary driver of therapy is the estrogen receptor status of the disease. So when we look at the journey of a breast cancer patient, we typically diagnose these patients with biopsy, where we take a little bit of tissue from a tumor. we analyze it, and there's a lot of things that are identified. There's different genes, there's different receptors, but by incidence, about 80% of breast cancer is estrogen receptor positive disease. So the primary therapy there is to suppress the estrogen receptors, and it's been tremendous. When I first started nuclear medicine, breast cancer patients would show up for their bone, liver, and brain scan. We're talking mid-80s. They come back a second time looking sick from chemo. They come back a third time and they didn't look so good, and we typically didn't see them a fourth time. Then the drug called tamoxifen came out and changed all that. You know, these patients take a pill. They don't get sick from the pill. It's like taking a thyroid medicine or taking birth control. You pop the pill, you have your coffee, and it treats your disease. 80% of breast cancer patients have long-term durable response to estrogen suppression therapy. About 30% of them during the course of their treatment, they progress. The question on progression is always, is the tumor, is the disease insensitive to that therapeutic product? And there's multiple products that can achieve that response. And again, we go from the initial drugs that are like Walmart drugs, $50 a month, onto $500, $1,500. Newer therapies are over $15,000 a month, and we have a 40% failure rate at 18 months. But the question is, these patients progress, is the tumor not responding to that therapy? Or is the tumor lost its estrogen receptors, meaning that that tumor no longer is responsive to it? So you can give the patient any type of estrogen suppression therapy and it'll keep on growing. And the only method that we can use today to assess that is biopsy. Challenge with progression in breast cancer is patients usually, by the time they show evidence of progression, it's not one or two tumors, it's 5, 6, 10. Now, I use the analogy, if I have a room with 10 people in it, one person walks out, they have blonde hair, blue eyes. Is it a safe assumption for me to say the other 9 people have blonde hair and blue eyes? I don't know. So really what we're looking at is it's a statistical sampling error when we do have biopsy. We're making an assumption that all tumors behave the same. And published data shows that somewhere between 20 and 40% of these patients that are thought to be by current means estrogen receptor positive disease actually have some component of negative disease. So it answers a question that's being asked, but for lack of a better tool, you know, we biopsy what we can get. And if we can't get tissue, if it's lung disease, if it's deep tissue, if it's bone, patients keep on getting treated based on the available genetic data. And then, you know, as they don't respond to other diseases, then the patients will eventually progress onto chemo. So for us, this gives availability of data to the doctor to potentially make a decision much earlier and drive the patient to a more appropriate therapy, you know, much sooner during the course of progression.
Maybe it's because I'm not a physician, but can you explain just exactly what's, and maybe you did and I missed it, but what does Cerianna do exactly?
So it's a radio-labeled version of estrogen. So what it does is it actually attaches to the same binding sites on the cells that these estrogen suppression chemotherapeutics target. So if all the disease in the patient lights up, we know that it's all estrogen-driven disease, that they're just, you know, breaking through, pick a drug, an aromatase inhibitor, whatever they're on. If we see tumors that don't pick up the estrogen, the radio-labeled estrogen, means that they no longer will respond to estrogen therapy. And then those are usually the cause of progression.
So you use a different therapy.
Yeah, it drives the therapy down a different path.
So the purpose, so the goal of Cerianna is to help the physician or understand the proper treatment for the patient.
Exactly.
And does Cerianna apply to all patients or only patients with estrogen related?
So our label is silent to ER plus or minus patients. Our label is for the detection of estrogen receptor status in recurrent and metastatic breast cancer.
And what percentage uh, of breast cancer is recurrent or metastatic?
So 30% of patients progress. Right now, we're looking at prevalence of over 200,000 patients.
Right. And how does the doctor know that Cerianna is the appropriate biomarker? Or is it an automatic test, an automatic uh, treatment or test that they perform?
So that's part of our challenge. This hasn't been widely available. The drug was invented decades ago, but the lack of a commercializable formulation hasn't really gone very far up until now. Um, we've been talking to oncologists and imagers all over the country, and pretty much unanimously they can see where they can position it in their clinical routines today. The key thing is 80% of patients are on estrogen therapy. As they progress, the question is, again, is that tumor still the same?
I see.
Yeah,
So,
this isn't expected to replace biopsy by any means. This is an adjunct. I would even argue that, if you needed to get tissue, it would be great if you could identify the tissue that isn't like all the rest. You want to look at that discordant tissue because that genetic data will give you a lot more information on the appropriate types of therapy.
I see. Well, so smarter people than I will know how and when to use it.
Smarter than me as well.
Uh, Now, what's the half-life?
So it's 110 minutes. It's similar to FDG as far as distribution strategy.
I see.
We can distribute it, several hours from the manufacturing sites. We have currently on our label 10-hour stability. We will be submitting for 12 hours, 12-hour expiry next couple of weeks, I believe. So when you look at it from a distribution strategy, we're virtually identical from the standpoint of fluoride deoxyglucose.
So, Peter, how many SNMs and SNMMIs have you attended?
I've been going to them since 1987.
Right. When was the first? Do you remember? I don't know myself.
I think Washington, D.C., one of the first ones.
Well, as I said before, you are an extremely innovative guy, and over the years, I've learned a lot from you, and you've contributed a lot to our publication, for which we're very grateful.
You guys have been to me as well, and I've learned quite a bit from you.
And you're a hell of a nice guy, and I'm very glad that I know you, and thank you very much for joining us today.
Thank you very much, Phil.
Keep us up to date on Sariana and how it progresses. Maybe we'll come back again in a year or 9 months and see how it's going.
I would absolutely love the opportunity.